Okay, so to wrap up our conversations about DNA and how it works, so um, make sure that you're really, really clear on the process of protein synthesis before watching these, because it's really important that you understand like the basic mechanisms of how DNA codes for RNA and then how RNA codes for a protein. Okay, so that just sort of that basic process of transcription and translation are really important kind of starting point for all of this. Okay, so we're going to break this up into two sections. We're going to talk about gene expression and mutation and cloning. That's going to be one section. And then we're going to talk about some other biotech stuff, some other biotechnology topics. The goal of all of the vast majority of this section of the chapter is just to kind of appreciate, at least for the biotech portion, is to appreciate the possibilities of what we can use DNA for as far as technology, right? So it's going to be sort of superficial. I'm going to talk about some things that aren't really in your book very heavily. Okay, so anyway, you've been warned. Okay, so like I said, you got to have the protein synthesis stuff, at least you need to be comfortable with that before we get too far into this. Okay, so with that, let's start. Let's talk about gene expression to begin with. So one of the things that I remember being sort of the most confusing thing for me as a beginning biology student, and remember, my tendency, my personal affinity, right, is more for big things biology, right? I like ecology, I like whole organisms. I really like, animal behavior. I'm really interested in how different organisms interact with each other. That's like my jam, right? Um, I'm super fascinated by all of this tiny invisible, as I call it. My friends, I joke around my friends about how they're all into invisible biology. Um, that stuff's fascinating. I'm really interested in learning about it, but it's much harder for me to kind of wrap my brain around. And so one of the things that I remember being sort of confused about in high school and when I first started taking college biology courses is this idea that we talk about how in mitosis the result is you end up with genetically identical cells. Well, the way that I had internalized that is cells that are identical, right? And that's not what it is, right? So genetically identical just means they have all the same DNA. It doesn't mean the cells are the same, right? And it took me a while to kind of, right, to kind of make that distinction. And so let's start there, okay? So you have all different kinds of cells in your body, right? So many different kinds of cells. They all have the same DNA, unless we're talking about your gametes, then they don't. Okay. But every other cell in your body all has the same instructions, right? They all have the same DNA. So how do those cells end up so very different from one another, right? That's the kind of interesting part. Um, and what it has to do with is it has to do with whether or not different genes are active. Okay. So just as an example, right? Here's intestinal cells, here's the nerve cell, here's a white blood cell, just as some examples. These are all cells that are found in your body, right? And here's a bunch of different types of genes, okay, that may or may not be activated, right? So the gene for glucose digesting enzyme, right? Well, all of your cells need to be able to utilize energy. So all of them have to have that gene be active, be functioning, right? To make an enzyme that can break down glucose, because that's how you do cellular respiration. Yeah? Okay. So everybody's got a check mark with that one, right? But antibodies are a thing that is a job of your immune system, your white blood cells. So as you can see, the antibody gene, right, is only checked here in our white blood cell. So nerve cells and intestinal cells, while they have the instructions, because everybody, all of your cells in your body have the same instructions, that particular gene is not active. It's not used at all, right? It's there, but it's nobody's reading it, right? Okay. And then our lactase gene. Oh, lactase. This is another good example. of an enzyme, right? Remember that any word that ends with ASE is probably an enzyme. And lactase is the enzyme that is responsible for breaking apart lactose. This has come up a few times now. So anyway, so in your intestines, right? In your stomach and your intestines as well, mostly your intestines is where that is happening. Yeah. Um, Hemoglobin gene. So hemoglobin is the protein that carries oxygen in red blood cells. Notice it's not active in any of these three because that's not the job of these cells, right? So your cells, even though they're all genetically identical, they have the same DNA, different parts of the genome, right, of the collective sum of all of your genes, different parts are activated in different cells. And that's how you end up with different cells, right? Also, different parts can be activated at different times. So not only do different cells produce different proteins depending on what genes are active, but when they do it varies quite a bit as well. So there's no point in making lactase, for example, unless you are consuming lactose. Otherwise, it's just a waste of energy. I don't know if you guys can hear that, but the ice cream man is driving down the street. Speaking of lactase, the ice cream man, very ironic. Anyway, right? So it would be a terrific waste of energy to be making a ton of enzyme that breaks down lactose if you're not actually consuming any lactose. Yeah? Okay, so all of this stuff is kind of variable. The process by which this is controlled is really complicated. So the thing I want you to know about this is not necessarily like the details of like, here's all the different, ways that this happens, that this is controlled. Simply just that there are a lot of different places in the process where things can sort of get turned on or off. Okay. So that's kind of the way I like to describe it. So I like to say that when a gene is being transcribed and translated, I like to just kind of say that gene is turned on, right? And other times that gene is turned off, meaning it's not being transcribed and translated. Okay. That's a little bit of oversimplification, but once again, that's the level of detail that we need here. Okay. All right. So if you're looking at this picture, some of this might look familiar, some of this might not, right? So, oh, look, here's our DNA at the top, right? There's our little DNA. Here's our gene. Oh, remember those whole intron exon thing, right? So the introns are removed. Okay. We'll talk more about that in just a second, right? And then, oh, there's our little guy, right? Leaving the nucleus. And there's our little mRNA. Oh, and it can be broken down. Oh, that's interesting, right? Or it can be translated to make a polypeptide, and then those can be broken apart or stuck together or blah, 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 blah. Okay. So this is our general pathway that we've been talking about. Okay. And each one of these little, they look like little faucet, like spigots, right? Each one of these represents a place where you can turn it on or off, right? So you can adjust whether or not transcription is happening or not. You can adjust how the splicing goes down, right? You can turn translation on or off. Okay. So like I said, I don't want you to like memorize, there's these 1, 2, 3, 9 different ways. I don't know how many there are in there. I didn't even count, right? I just want you to have an idea, have the idea that there's lots of different ways. And then we're going to talk about a couple of things. Okay. So one thing to know about are these things called transcription factors. You don't need to know the details about this, but essentially what transcription factors are is these are proteins that essentially allow for transcription to occur. So you remember this, you remember RNA polymerase, right? It's shown in orange in your book and in the other PowerPoint that we looked at. And that's the actual enzyme that is making the RNA. Yeah. Well, it turns out it doesn't just like jump on there by itself. There's a lot of other structures that are associated with getting it into place. Okay. Called transcription factors. All right. So like I said, not a ton of detail there, but this is one of the ways that transcription can be controlled, either turned on or turned off. If these proteins aren't present, transcription is not going to happen, right? If they are, it is, right? So this is one way of controlling whether or not transcription occurs. Another one of the things from that long list is alternate RNA splicing. And this one is really, really interesting. So you remember the different introns, right? Remember, intron stands for internal non-coding region. And those are the little areas that, like I said, some people think of think of it as junk DNA, that are cut out, right? So they're removed because they don't have a part of the message that is important to be translated later. Well, what's interesting is that some stretches of DNA have different, under different circumstances, you can remove different parts, right? So this is saying, all right, so we've got these areas, right? Little segments, 1, 2, 3, 4, 5. And all of these introns are the tan, and they're all going to be removed, okay, regardless. But another thing you can do to sort of manipulate this is you can remove some of the exons as well. So if you also remove 5, you get 1, 2, 3, 4. Or you can remove 3, and then you get 1, 2, 4, 5, right? So another way that you can end up with multiple possibilities from one strand of DNA is by having various different ways that the RNA can be spliced out, right? So that's kind of interesting as well, right? So even a single gene of DNA can produce multiple different mRNAs depending on which pieces are removed. Okay. All right. So that is like just a couple of examples of ways that genes can be turned on or turned off, right? Their expression, gene expression, can be controlled. Okay. Another thing that I want to talk about a little bit, without getting into it too much, is another really important thing to realize is a lot of people, you come out of the last lectures going, okay, I understand this. We have DNA and it makes some RNA, and then that RNA is used to make a protein, and that protein does something. So it's an enzyme or it's a whatever, right? It's a protein that does a thing. Okay. But there's lots of genes that control other genes. Okay. And one really interesting big picture way of thinking about that is to talk about developmental genes. Okay. So the other, there's kind of multiple names that we can use. Okay. When a multicellular organism grows, there's this issue of not only which genes are activated, are expressed at any given time, right? And in what kinds of cells, right? So your cells that are going to become red blood cells are not going to express the same genes as your genes that are as the cells that are going to become bone cells, right? Back to the first image in this PowerPoint. Not only that, but also there's this organization of your body, right? Where you have different parts in different places, right? You have a front, you have a back, you have a top, you have a bottom, right? You have this three-dimensional arrangement that's really, really important. Okay. So it's really, really important that you're not growing a finger where your eye is supposed to be, because that's not going to help you see. Fingers are very useful, but they're not really useful there. Okay. So in addition to just this idea of we have different types of cells, it's really important that genes are expressed not just in the right kinds of cells, but that we have this cascade of where structures develop in your body. Okay. So without getting into the details of this, because this is way complicated. Okay. Without getting in the details of this, when an egg gets fertilized, right? So when that process of sperm and egg joining together, at that moment, the orientation of where different structures are going to develop in that zygote is kind of set in motion, right? As far as where's the front going to be, where's the back going to be. Okay. And so you totally don't worry about like all of the words on this picture, because look at it if you're interested, but don't feel like, I have to memorize this, because you don't. Okay. But the idea is that different chemicals, right, are present in the front versus the back. Different genes are being expressed in different places in the front versus the back. Okay. And essentially that cascade, that process says to your body, right, to your body, developing body tissues, this is where we grow our head, and that's where we grow our butt. Right? And you have different structures associated with your head and different structures associated with your butt. Okay? Sorry, I'm waiting for an important phone call. I had to check. Right? So, yeah. So you have this like layout of where things are going to grow. Okay. And so the genes that are responsible for this are called developmental genes, or more specifically homeotic genes, or simply Hox genes. That's a bunch of different names that for our purposes essentially mean the same thing. Okay. And so here we're looking at the development of a fruit fly, because fruit flies are used a lot for genetics research. They're small, they're easy to grow. Okay. And so this is kind of showing in a graphical nature, it's kind of showing that process of how does the front know it's the front, right, in a fruit fly. And it's a very similar process for you, actually, right? So here we're comparing the homeotic or Hox genes of an insect, a fly, with a mammal, right? A mouse in this particular case, right? So you're mammal, you are a mammal as well. All right. And so the idea here is that even though the details are different, right, the set of genes that is responsible for determining sort of which end is the front, which end is the back, is actually very, very, very similar between all animals, right? What the genes do and how similar the genes are, shockingly similar, right? Even in things as different as, say, comparing you to a sea anemone, right? Or a sea jelly. Yeah. Your layout and their body layout are controlled by genes that are incredibly similar. They haven't really changed that much, not as much as you would expect based on how different you are from a jelly, right? They haven't changed that much throughout evolutionary history, which is kind of interesting. Okay. And so one of the things you'll notice right away, this is meant to show you like a strand of DNA with different genes. And then the different colored bands are trying to show you, this gene is responsible for the development of mouth parts, right? This gene is responsible for the development, the green one, for the development of like the head and the eyes in the fly, right? And then you can see where they work in the mouse as well, right? So the same like color coding. You'll notice there's a duplication. So one of the big things that happened in mammals is invertebrates in general, in all animals that have a backbone, is this duplication, so that we end up with a lot more copies of those genes, right? So the DNA got copied. Okay. I don't want to get into that's a path that's a rabbit hole that we could go down, but let's not. Okay. So these genes are really, really interesting. And the way that we learned about these genes actually is by studying mutations in these genes. So one of the things we're going to talk about next in a couple minutes is we're going to talk about mutations. Essentially what a mutation is any change in DNA. Okay. And so fruit flies, like I said, fruit flies are a great example of an organism that we can easily manipulate to see what happens when we mess with their genes. Okay, so you can cause mutations a variety of different ways. We'll talk about a more extensive list in just a second, but one example is with ultraviolet radiation. Okay, and so if you blast a bunch of fruit fly eggs with UV radiation or other types of radiation, right, you can cause all kinds of mutations and then basically just see what happens. Right. And so here are a couple of fun examples, right? So here is one example where this is sort of the normal fruit fly body, right? Notice it has red eyes. Notice it has one pair of wings, right? So that's typical. That is the typical arrangement of things in a fruit fly. This guy, right, has a mutation where he has 1, 2 sets of wings. And the reason that he has 2 sets of wings is a mutation or a duplication in the gene that is responsible for that segment of the body, right? That he gets extra wings. He also, it looks like there's some other mutation in his eyes too. It looks like he's got an eyeless situation as well, because his eyes don't look normal either, right? But that's really interesting, okay? And then here are some other examples of cool things happening in weird places. So there are gene mutations that we've discovered that can cause basically any part of a fly to grow in the wrong place. So this little fly here, this poor creature, right, instead of having little antennae coming out of the front of his face for sensing things in his environment, guess what? He's got legs growing out of his face. And look at this guy. On his leg, he's got an eye growing. Yeah. So these are examples of genes that control not if something develops, but where they develop. Okay. So in addition to this idea of, being able to turn genes on and off, right, to allow certain things to be produced or not, right, like depending on what kind of cell you have, right, there's also this issue of where that matters, right? So you have certain body parts in a place because that's a place that makes sense to have them, right? Or at least that's the result of the evolutionary processes that produced us. Okay. All right. So those are Hox genes or homeotic genes. Don't worry too much about any sort of technical details of that. Just kind of have an understanding of these are the genes that are responsible for location, right? That you grow eyes where your eyes belong, right? That you grow fingers where fingers belong, that kind of stuff. Okay. And those are shared genes in all animals, which is really, really cool, right? Okay. So, like I said a second ago, we have to talk about mutations a little bit more. Okay. So, a mutation, I'll even write this down. No, because my pen's being a jerk. No. Okay, so any change in the sequence of DNA is a mutation. All right. And this graphic shows you some examples of mutagens. So a mutagen is anything that can cause a mutation. So what do these pictures represent? So this person is getting x-rays. So high energy radiation is a mutagen. It can cause DNA to become mutated. Okay. And then they have a beach scene here. So UV radiation, basically any of the high energy types of radiation, like ultraviolet radiation, X-rays, gamma rays, any of those things can cause mutations. So we consider them to be examples of mutagens. And then here's this beautiful lady smoking, right? And so there are chemicals in tobacco smoke that are mutagens that cause cancer, right? And this is, why lung cancer is a thing with people that smoke. But, and I always want to point this out that anytime you're inhaling any kind of burning plant material, not just tobacco, but any other kind of burning plant material, whether it's weed or whether it's a campfire, right? Burning anything has chemicals in it that are hazardous, right? You don't typically hear about people developing lung cancer from, a campfire because you're not habitually inhaling large quantities of campfire smoke, right? But it still has those chemicals, okay? And for that matter, vaping hasn't been around long enough for us to kind of fully understand the details of what vaping does. many people, well, there's some evidence to suggest that vaping is slightly less hazardous in some ways, because it reduces cancer risk. The problem with vaping, though, is depending on what you're inhaling, a lot of times there are all kinds of chemicals in the vape juice that are not healthy, right? So it has all kinds of stuff in it, preservatives and other things to add flavor and, you know, all kinds of other stuff that is that's not good. It's not good for you to inhale. Okay. So while a lot of people choose to vape instead of, you know, smoking cigarettes or cigars or whatever, right, as a way of getting their nicotine fix, there are other risks associated with it. Okay. Something to weigh, right, and think about. Okay. So any kind of burning plant material, right, has potential, or any kind of burning material at all has potential to have mutagens. And then there's other environmental factors as well, right? Certain types of foods um, have higher risk of causing mutations to DNA than others, right? So if you have a really high fat diet, that increases your risk of certain types of cancer, like colon cancer. Yeah. So there's all different things in your environment that are mutagens. So anything, one sort of good rule of thumb is anything that can cause cancer, right? Um, can be considered a mutagen, because the way that cancer is often triggered is by mutations in the genes that regulate cell division. And so your book talks about this in a little bit more detail than I'm going to. If you're curious about it, please read, but I'm not going to get into it too much more than that. Okay. So that's a mutation. And then I don't know why I always show this. It cracks me up. It's so funny, right? Here's a bunch of images of different fruits and roots and different plant parts that look funky, right? And so all of these have had some sort of mutation in their developmental genes that control how they develop, right? So instead of, right, developing a single cherry, here we've got a cherry with a weird nubbin. Okay. There is nothing dangerous about eating this cherry. It's perfectly healthy to eat, by the way. You can eat weird looking fruit. It's not going to make you sick. Right? It just means that instead of just developing one, it developed one in a nubbin, right? Or the squash here didn't really develop one squash. It's kind of like a twofer. Yeah. This kind of stuff happens, and it's just sometimes it's a result of some sort of weird blip in the developmental genes. Sometimes it has to do with the environment that they're growing in. So you can get all kinds of funky shapes in carrots if you try to grow carrots in rocky soil, because the way that they grow, right? Carrots, the root is growing down, 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 down. And if it runs into something, guess what happens? It can't grow through it, right? So it'll split and grow around it, right? Or it'll bend. Yeah. If you plant your carrots too close together, sometimes you get carrots that are like twisted around each other, because as they're growing, they run into each other, right? And that changes their shape. So some of this is a result of, some of these examples are a result of growing conditions that have nothing to do with DNA. It just has to do with getting in the way of things, like this weird potato, right? Probably was just an issue of like there were rocks or roots from another plant or something in the way, and it just grew around them, right? Whereas others, yeah, are, you know, some sort of just funky developmental thing. Okay. So that's just kind of, I like that picture because it's just kind of fun to look at. Okay. Okay. So more about mutations. This is actually something that we're going to be exploring in the lab that's associated with this. Okay. So this week, we're doing a lab where you're going to be exploring a couple of different types of mutations and what the effect of those mutations are. Okay. So there's more than this, but there's two main categories of types of mutations. Okay. And we call these point mutations and frame shift mutations. Okay. Um, in a general sense, a point mutation is where you are swapping out you're swapping out one or more nucleotides for a different one. Okay. So, where you had an A, instead it's a G, right? So it's just a change in, sometimes it's only one nucleotide, sometimes it's a couple nucleotides. Okay. So that's called a point mutation, usually because it happens at one point. Okay. A frame shift mutation is called that because what you're doing in a frame shift mutation is you are either inserting, as the name insertion applies, or deleting portions of DNA. Okay, so inserts or removes nucleotides. Okay, whereas in point mutations, you're just swapping it out, right? So the A becomes a G, I think is what I said the first time I said this. In frame shift mutations, what's happening is you just take that A out completely. Or where the A was, you also add in a G. So like you shove a G in between the A and whatever was next to it. Okay, so why are they called frame shift mutations? We'll get to that in just a second. Okay, but the way I want you to think about them is when you go to like a drugstore, okay, and it depends on the particular, what comes in this kinds of display varies, but I always think of like like you're at Walgreens and you're looking at lipstick, right? And or deodorant sometimes is displayed this way, and it's in like a line, and if you take one out, the whole thing like slides forward. And then if you're like, oh crap, I don't want that, you got to like push the other ones back to shove it back in, right? Right? Because it's not just sitting on a shelf, right? It's like in this like spring-loaded thing. Okay. That's what I always think about when I think about a frame shift mutation, because essentially the idea is if you remove a nucleotide, guess what? Everything else fills that space. It's not like a space is held open. It's not like, oh, there's no gap and it, whatever, right? The space disappears. Everything moves forward if there's a space, right? Or if you insert something in there, right? Let's say you're a jerk and you decide you want to mix up all the lipsticks. I'm going to take, there's these 2 different colors of lipstick. I'm going to take all these, I'm going to shove them in the other one, right? And so what you're going to do is you're going to put them in there and it's going to push them all back, right? And so that's what would happen with an insertion, right? Is it would screw that up. Okay. So we'll talk about why they're called a frame shift in just a second. All right. So what I want you to think about for now is, just as far as changes go, which seems like it might make a bigger change based on what you know about protein synthesis? Which one seems like it would make a bigger change? Swapping one nucleotide, or even a couple nucleotides, but just swapping one out, or removing something or inserting something? Which do you think is going to typically, not always, cause a bigger change, a more profound change? Okay, so just kind of think about that for a second. All right, so we're going to talk about different types of point mutations as well, right? So all I did here was I defined what a point mutation is. Okay, so now what we're going to do is we're going to talk about what a silent mutation is, a missense mutation is, and a nonsense mutation. Insertion and deletion are exactly what they sound like. So you're either inserting or deleting, right, nucleotides. Okay, so silent, missense, nonsense. This little graphic discusses that. Okay, so a silent mutation. I can't write small enough. I'm just going to go like this because I'll scribble all over it and then you won't be able to read it. So let's just say here's our original DNA sequence, ACA. Okay, and we're looking at our different types of point mutations. So remember, now we're just talking about these. We're not talking about those. Okay. All right. So whoa. Okay. So our original sequence is ACA in this particular example. So if there's no mutation at all, the ACA is going to code for UGU, because remember, A codes for U, C codes for G. Okay. And then remember, you do not need to memorize this. But if you were to look up ACA, or excuse me, if you were to look up UGU on the genetic code table, either the circular one or the square one, it doesn't matter which one you use, from the last chapter, right? Or not the last chapter, same chapter, but the last lecture, okay? You would see that UGU is the codon. Remember codons, 3-letter words? UGU is the codon that gets you cysteine. Okay. So a mutation is considered a silent mutation if it does not change the amino acid. Okay. And if you're, by the way, if you're suspicious of, if you're skeptical of all of this, you can get out your little genetic code and look at this. Okay. So if our original sequence is ACA and it is mutated, a point mutation in that last letter. So instead of ACA, now it's ACG. Okay. After it's transcribed, you would get UGC instead of UGU. But it turns out that UCG produces the same amino acid. It still makes cysteine, right? So they call it a silent mutation because it doesn't actually change the amino acid at all, right? So even though there's been a change in the DNA, the protein, the resulting protein isn't changed at all because the amino acid is exactly the same. So it's silent. It's a mutation, but it doesn't change anything in the end. Okay? All right. Missense. That's kind of a weird word. Okay. So what missense does is it changes it so that the result is that you end up with a different amino acid. Okay, so let's follow this one across, right? So if our original DNA sequence is ACA and there's a mutation that changes the A to a T. So now TCA is going to get transcribed into AGU. And then AGU is the codon that codes for serine in translation. So serine is not the same as cysteine. So we have changed our amino acid. That's what missense means, right? So this particular amino acid, instead of being cysteine, what it was originally, right, is serine instead. Okay. A missense mutation is different from a nonsense mutation because a nonsense mutation changes what was an amino acid into a stop codon. Okay, so let's say, right, our ACA is mutated to ACT. And notice for all of these, we're only changing one letter, hence the point mutation, right? We're changing one thing at a point. We're not inserting, we're not deleting. We're just swapping out one letter, okay? So instead of ACA, it's ACT. Well, that codes for RNA of UGA. And if you look up UGA on the genetic code table, you'll see that doesn't line us up with an amino acid. That is one of the 3 stop codons. So nonsense because it stops. Think of like non as in not going any further. Okay, so missense, I always think of missense as like a mistake in the amino acid. Missense is mistaken amino acid, whereas nonsense is a not going any further because it's a stop codon instead of a different amino acid. Okay, not going any further, nonsense. Mistake, missense. Silent doesn't make a difference. Okay, so those are the 3 types of point mutations. All right, now let's think about what the ramifications of those are. So when a protein, one of the things that we talked, and this was quite a while ago, right? So when we were talking about what proteins are, we talked about how they tend to be very large molecules. Not always, but many of them are quite large. And they, in addition to the sequence of amino acids, they have a very interesting 3-dimensional shape that has to do with the way that they all kind of, that polypeptide, right? That chain of amino acids folds up. Okay. If it doesn't fold correctly, remember this whole form follows function thing, right? If it doesn't fold correctly, the protein isn't going to work the way that it's supposed to. Okay, so let's talk about point mutations and how much they would affect the resulting protein. Okay, so silent mutations, we're not even changing anything, right, from the protein's perspective. So the protein is going to function exactly the same. Nothing is going to be different. Okay, how about a missense mutation? Well, it means that one of the amino acids is going to be different. Is that a big deal? It kind of depends. It depends on how different that amino acid is from what the amino acid should be. So if it's a really similar, if the structure of the mutated amino acid is similar to the structure of what the original amino acid should have been, maybe it doesn't do much of anything interesting, or maybe it changes the way the whole thing folds, right? And it makes the protein completely different. Okay, so missense mutations can have a range of effects. It can be very minor to huge difference. Okay, so, right, so silent is no effect really, no big deal. NBD. Okay, absolutely no change, right? Whereas serine can be like a range of no big deal all the way to like it completely makes it so the protein doesn't function the way it's supposed to at all. Okay. What if you have a nonsense mutation? What is a nonsense mutation going to do? Well, if you hit a stop codon, what does that mean? That means translation stops. So that means depending on where it's found, if it's at the very end of where the mRNA was going to stop anyway, it maybe stops like 1 or 2 codons early, may or may not be a problem. But if it's near the beginning, it's going to totally change the protein, right? So usually a nonsense mutation cause big changes, right? So usually that means big difference in your resulting polypeptide, your resulting protein, because essentially what you've done is you've just stopped translating way before you were normally going to. Okay? All right. So those are our different point mutations. Then we have a frame shift mutation. Okay, so I already talked about lipstick and the spring thing, right? Okay, so let's look at the outcome of a frame shift mutation. Okay, so they've included here, very conveniently, a little table, right? So that you can look this stuff up so that you're, right, you don't have to keep flipping back to your genetic code. Okay, and so what we're looking at on this side is we're looking at the original DNA sequence and then the RNA sequence that you would transcribe. Oh, look, AUG, remember that one? That's our start codon. Imagine that they've got that there in the beginning. That's fantastic, right? And then after translation, this is the series of amino acids that you get, okay? All right, so what they're showing you here, is this an insertion or deletion that they're showing you? It's an insertion because the arrow is in, okay? So it's like we've got that line of lipstick and we're shoving one in, okay? So here's why it's called a frame shift mutation. Okay, so notice how they've boxed this off in units of 3, because remember, codons are how many letters long? Codons are 3 letters long. Always. A codon is 3 letters, period. Okay, so what they've essentially done here is they've shoved the A in between that T and that C. Okay, so that bumps the C that way. Okay, so it was TCA, ACG. Now it's TAC, AAC, and then it was GAT, and now it's GGA. So essentially, we call this a frame shift mutation because the reading frame of the 3-letter codons is different everywhere after where that insertion or deletion. This is showing you an insertion, but a deletion would have a similar effect, right? Basically, it changes everything after that. And so if you can't, imagine that, just look at it in this example, right? It's showing you if we shove an A in right here, here's how the DNA is going to be different. And then here's how now the RNA is going to be different. And then that's going to make the Protein different too. Look, the first one is still the same because that didn't get changed. But from the point where the mutation was, I shouldn't say point, from the area where the frame shift mutation was, where the insertion happened, everything after that is different now. Yeah? Because we've shoved something in there or pulled something out. Yeah? So relative to our point mutations, now remember there was a bunch of different kinds of point mutations. What would be the impact of this, of a frame shift mutation on a protein? It looks like it's going to be a profound difference, yeah? Because it's not just changing one amino acid, it's changing like every amino acid, right? From the mutation point on down. So frame shift mutations tend to be more profound. Notice I said tend to, not always. Tend to be more profound changes when compared to, certainly when compared to a silent mutation or a missense mutation, right? Which are two types of point mutations. So frame shift mutations tend to be a bigger deal. Okay. I hope that makes sense. All right. So here's that business. Where do I want to? Yeah. Okay. Okay. I might, as I'm going here, I'm thinking I'm going to change my plan here for the videos. So here's the bit in your book that talks about mutations in cancer. So like I said, I'm not going to get into this too much, but the idea is that when we're talking about mutations causing cancer, those are mutations that occur in specific types of genes that are called proto-oncogenes. Onco means cancer, right? So when you have cancer, you see an oncologist, right? There's an that, right? An oncologist is a doctor who specializes in treating cancer. Okay. So a proto-oncogene are the genes that are normal, right? That are responsible for normal regulation of the cell cycle, right? Normal, healthy cell regulation. If there is a mutation in those proto-oncogenes, then they become oncogenes, which basically now those genes aren't working the way they're supposed to. And that's how you end up with the out of control cell division. Okay. So this just relates back to our, previous conversation about cancer. Okay. All right. The last thing I want to say about mutation, and then I think I'm going to cut this here and we'll talk about cloning with the next section. Okay. So mutations. Are mutations bad? Well, you might already have some opinions about that based on the discussion that we were just having, right? This idea that if you're, if we're talking about a silent mutation, is that good or bad? Well, it doesn't change anything. So it's neutral, right? Are they bad? Well, if we're talking about oncogenes, then yeah, they're bad. Right? So the idea is that mutations, I think people hear the word mutation and they think of one of two things. They either think, oh, cancer, bad, or they think of Marvel heroes and they're like, oh, the X-Men, they all have mutations and that's cool. I want a mutation because I want to be able to move metal with my mind, which by the way, mutations can't do that. Comic books lie to you. Okay. They have good messages about, humanity, but they're not real. Okay. Humans aren't going to mutate the ability to move metal with their mind or shoot laser beams out of their eyes or any of those other cool things. Okay. All right. Or, throw ice that they can then, surf on, whatever. Anyway. Okay. Back to my point. So are mutations bad? Yes. But they are also neutral. They have no effect at all. And for that matter, mutations are also good because, and this is why are we looking at this beautiful picture of a coral reef? Fundamentally, the only way to get variation, the only way to produce all of this variation in living things, right? To make all these different kinds of organisms, like 10 million different kinds of living things on this planet, is through mutations, right? The only way to get new organisms, new plants, new fungi, new animals, right, is through mutation. And some mutations turn out to be incredibly advantageous, right? So the mutation that allowed our brain case to stay really round, that allows us to grow big giant brains, was a hugely beneficial mutation, right? Because by having big brains, we're able to do all kinds of cool things. Yeah. And so mutations aren't always good, bad, right? It depends on what the mutation is. Okay. So yes, some of them can be harmful. Some of them are like a genetic dead end, right? They produce an organism that can't survive. Sometimes they negatively affect organisms that are already alive, like in the case of cancer or things like that. They can also be totally neutral, have absolutely no effect at all, or they can be amazing and produce all kinds of really interesting characteristics, right? So all of the cool things that your body can do, every single one of them, ultimately is a result of a mutation of some kind or another, right? The fact that we have super sophisticated eyes that we can use to see, right? Mutation. The fact that some people have curly hair and some people have straight hair, mutation, right? The fact that, you know, some people can bend their thumb at this joint all the way back and some people can't, mutation. I'm picking lame superficial things, but you get the idea, right? um, all of that stuff is related to mutations. So mutations aren't necessarily bad, they're not necessarily good either, right? They can be anything. Okay, so I don't want you to have like this negative kind of connotation in your head when we talk about mutations. Okay, we're going to stop here. And so the next video is going to be cloning and biotech. Okay, all right.